Thank you. Thank you for the invitation. It's great to be back. I've been back a few times since I graduated in 1999. But this is the first time I'm seeing the new fancy building you guys have. So, and I'm really glad for the invitation, and then also pushing me to come in person, because there was a time when I was thinking that I may not be able to do that trip, but then they really motivated me, that enticed me to come and see, so that I can see the building, which is totally worth it. So, and again, and I think, like, I see, it's really great to meet a lot of my old friends. I'm sure some of you are also in the audience in the Zoom, which I have not met, but it's really a pleasure to be back here. And so today the topic is, like polygenic risk score, which is an area I've been working on for now more than a decade. And this is an area, it's very interesting history. Like, when the Genome-Wide Association study just were sprouting, people are very excited about it. There was a, genetic variants started emerging that associated with many disease. But then the enthusiasm went down, because there was people at some point thought it's not going anywhere. And then suddenly in the last 3, 4, 5 years there's a lot of excitement because of the large Genome-Wide Association studies. So today we're going to talk about different components of it, different aspects of the thing, starting with a little bit of the design of this kind of studies in terms of sample size requirements, as well as some of the algorithms that is happening, to develop better polygenic risk scores, and how this could be made more fair in terms of their performance in different ancestry populations. And then at the end, I'm going to talk about some applications of these polygenic risk scores, potential application, and evaluate their potential utility. So just to orient people, if you're not familiar with the genetics, so just a very basic background. So we are looking at inherited genetic variations, that are genetic variations that we get from our parents and that are passed on from generation to generations. And we are looking at the very simple changes like single nucleotide polymorphisms, which are the most common type of genetic variations we see in the genome. And these genome-wide association studies that are using these kind of cheap technologies, where they can measure hundreds of thousands or potentially millions of these single nucleotide polymorphisms on relatively inexpensively on large number of people. And then basically with that, after doing some quality control, and you can use genotype data, you can use something called imputed data. That means using the genotype data on the SNPs that are in the platform, you can actually impute genotype or probabilistically on nearby SNPs, which are in correlation with the SNPs which are genotyped. And that's what you get the data, and then you basically run a lot of association tests with whatever phenotypes you are measured on that sample. It could be a case control phenotype or a continuous trait. And the first set of analysis is very simple. You run millions of regression. It could be linear regression, logistic regression, or regression based on Cox model. But then, what get really fun for statistician to work on is something called post-GWAS analysis. After you have done that initial analysis and made some discovery of the top regions where you see the strongest signal, but there are a lot of things you can do with this kind of large data set, and that's called post-GWAS analysis. So this is your basic data structure. If you had access to individual level data for each individual, you'll have genotype data, where a genotype is a combination of alleles, SNP allele they can carry in the 2 chromosomes they have, and that could be coded for numerically in terms of how many variant allele one person carries in each of these SNP locations. So it could be 0, 1, or 2. If you, for example, in the 3rd locus, if you think about count the number of G alleles an individual carries. So it could be the first 0, the second 2 individual is 1, and the third one, 4th one is 2. And the basic GWAS analysis is very simple. Like for each of these SNPs, you treat that as a X, which is like coded as 0, 1, or 2, and then you run a regression on Y after adjusting for some covariates, and you are focusing on this association coefficient, the association between the SNP genotype and your outcome, and you have that for many SNPs, potentially millions of SNPs, and that is considered sometimes the summary statistic data. That means after you have, instead of the individual level genotype data, you run the models for each individual SNPs and summarize the association coefficients, and those data are much more easily available. You can often download this kind of data from large consortium without signing a single paperwork. Whereas if you want to get individual level data, it's much more complicated. So a lot of the methods you will see that are being developed in the GWAS thing, like, that they say, hey, what can I do with the summary statistic data that I would like to do if I had individual level data? And it turns out that because we are working with mostly additive model, you can do a lot of stuff with the summary statistic data. So just to give a background, so like, it's useful to focus on a disease, like what has happened in the last 15 or 20 years since the GWAS era came. Before the era of genome-wide association studies, for a disease like breast cancer, we already knew the existence of genes like BRCA1 and 2, which contains these mutations, which are rare high penetrant. That means they are in the population, they're very rare, like, frequency is less than 1 in 100 or 1,000. But the woman who carries those mutations, they have a high risk for, let's say, breast or ovarian cancer, and even men can be at high risk for prostate cancer if you carry these mutations. And these were typically identified through linkage studies of highly affected families. But since the era of genome-wide association studies, like, initially the genome-wide association study started with few thousand samples, and then it kept increasing to tens of thousands. Now they have a study of more than 100,000 cases and controls, let's say for breast cancer, and you can see how the discovery has increased over time. Currently, we have known about 180 or so regions in the genome, each of which contains one or at least or even more than one susceptibility. This susceptibility variant, that means they're associated with breast cancer, and they're kind of independent because they come from independent regions of the genome. So with that context in mind, so what is the polygenic risk score? So you can think about just as it's a construct. So you can compute this quantity for each individual. And it's basically, you count the number of risk alleles one individual carries over these multiple genetic variants. And we'll talk about how do you decide that what needs to include in the model. You have a question? And how do you, and then you weight them according to their effect size. So that is and we'll talk about how do you estimate those effect size, but this is basically the construct of what is considered polygenic risk score. So the information on that, what is the effect size and how many SNPs you're going to include, they're coming from those genome-wide association studies, right? But once we have those being given, you can evaluate this quantity for many, many people in a new sample. So just to, the next few slides is a little bit about the design, like, what kind of sample size we need to have this genetic risk score to have good prediction performance. And it's really useful to think in terms of like that kind of the sample size requirement and how this concept relate to heritability. So if you take this polygenic risk score for any disease, and suppose the construct has been given, and you evaluate it a lot of people in a random sample of people in the population, it's going to start looking like a normal distribution. And the reason it looks normal distribution, although genotype data are categorical, but because you are doing a linear combination of many of them, and they're kind of independent, there's a kind of a central limit theorem is playing around, and it starts looking like these bell-shaped distributions. And what is a very critical factor is that the spread of that distribution, like how widely that the variance of that distribution is, and that kind of determines that how well this score will perform in terms of discriminating, let's say, cases and controls of a given disease like breast cancer. And there's one little nice result, that I find it very useful to think about how risk score performs in discriminating cases and controls. So if you now look at the distribution of the risk score, the polygenic risk score in a sample of cases, and then what you will see is that the distribution will be now shifted more rightward, because of course cases will have a higher value of the score, and you can show that by if the genetic score is well calibrated, and here I'm thinking about modeling risk in terms of the log risk scale. You can show by a simple Bayes theorem, if the original risk score in the population was normal, then in the cases, it will be another normal distribution with the same variance, and the mean shift is going to be exactly the same as the variance of the original score. Okay, so this is a simple, if you just a Bayes theorem, you can prove this. And why is that important? And the fact that, how well the distribution of risk score in the cases versus control is going to determine how well it can do in terms of for example, discrimination, like of the performance of the model. So what I told you right now, that if I know the spread of the original distribution, and if the risk score is well calibrated, that means in the sense that those parameters that are estimated of the SNP association coefficient, they're unbiased kind of, then I already know that how well that score is going to perform, because there is an exact one-to-one relationship between the spread of the original distribution and how much shift I expect to get between the distribution of cases and controls. Now, why is that all relevant? Because that goes back to the heritability of trace. So the performance of the PRS actually depends on a few factors. And one of the most important factors is a concept called heritability. And for the purpose of this talk, you can think about heritability for a set of SNP is the spread of that polygenic risk score that you can define based on the SNPs and the ideal polygenic risk score I can construct. That means if I had exactly known what is the true effect size. So right, that is the best polygenic risk score you can get based on a set of SNPs. And because and you can actually estimate this quantity that for a set of SNPs, small set of SNPs or a set of SNPs in the whole genome, you can estimate these concepts of heritability. That means basically again think about if there is an underlying true polygenic risk score, which is basically can be obtained if I knew exactly the coefficient of each SNPs, then the variance of that quantity it would be the heritability of the trait. And the way I am defining heritability, it actually relates to the more traditional concept of heritability, like for example, if you run mixed model in the GWAS, like and estimate heritability that way, they are just one-to-one transformation of each other. And I like to think about heritability in terms of the variance of that underlying true polygenic risk score, because that kind of relates to the clinical utility of the PRS, like the way I talk about this, it gives you the separation of the cases and controls. So you can think about, so the heritability kind of gives you the limit of what these polygenic risk scores can do, because that's the best polygenic risk score I can ever hope to get. But in practice, I cannot achieve that performance because I actually have to estimate those SNP coefficients from a given data. And that estimation is going to be always noisy. So we expect that those polygenic risk scores I'm going to build from a real data, they're going to be performed less well compared to the ideal place where I might want to go on there. And how fast, that will increase as the sample size increase depend on few factors. And we have studied and we have developed theories, and other people have developed theories that shows that depends on what is considered the parameters of the genetic architecture of the trait in terms of how many genetic variants are there, which is contributing to the heritability, and what is the distribution of that effect sizes of those traits. And those all determines, and I'll give you some specific example that how fast this performance of the polygenic risk score will increase, and when it is going to reach the limit that is indicated by heritability. So there are quite a few work, going over, from the time, like I like the first paper in this area, I think that explicitly connects the concept of heritability to utility of risk prediction is the Paul Farrell's paper. We have done work in this area, and I have been interested in this area since 2010, where we basically using some initial result from genome-wide association study, we try to characterize what is the effect size distribution of genetic variants for a complex trait. And then we could relate it that showed that if you can estimate somehow the effect size distribution, you can basically relate that to figure out the yield of future genome-wide association study, that how many more discoveries you can have as I increase the sample size, or how well the performance of the genetic score is going to improve over as sample size increases. And in 2013, we did a paper where we actually explicitly wrote down some theory that in terms of what is the performance of this kind of polygenic risk score is going to be as we increase the sample size. And we showed that basically the expected performance of models can be written theoretically in terms of the sample size of the training data set and those parameters of certain parameters of the genetic architecture of the model. And then that means if I can have some estimates of the effect size distribution, we can plug it in this formula, and then we can project it out and say like how as the sample size increases, what do we expect to happen in the future. And then the most recent paper, we did some work to actually estimate those effect size distributions more rigorously based on current studies, and so that we can make better projections in the future. So here is some examples. So that work, that culminating in this paper recently. So basically, we collected summary statistic data from about 14 different cancers, and then we using the method that was developed by my postdoc Dora Zhang, and in the 2018 paper, we tried to estimate for each cancer how many common genetic variants, the SNPs are there, that might be contributing to the heritability. Okay, and there is some underlying model and techniques. I'm not going through that. And so that on the second column, you see that for each cancers, across cancer, we estimated every cancer is very polygenic, right? That every cancer is associated with not only dozens or hundreds, every cancer is potentially associated with thousands of these common genetic variants. And on the next column, what I'm showing is the heritability associated with this variant. That means, remember, if the way I define heritability is the best PRS I can ever hope to construct based on those SNPs. And that could be, that also comes out as our modeling exercise, that heritability. So you can think of those as some variance parameter, but they're not scaled so that it has to be a number between 0 and 1. So it's not scaled to be between 0 and 1. And the final column, what I'm showing is that as I said, if I know the spread of that polygenic risk score distribution, I can convert that to a kind of how well that will give me the separation in terms of the cases and controls. So I'm characterizing that in terms of AUC. So you can see that as the spread for some cancer, the spread is high, like, for example, CLL or testicular cancer, the variance is high, and that you can see that the area under the curve is also high, like higher than 80%. And then there are some other cancers where you can see that the spread is low, like, for example, ovarian cancer, then the area under the curve is 64%. So you can think about the last column is basically gives you the limit. That's the one day, that's the best we can hope to achieve based on the polygenic risk score for prediction of these cancers, right? But this is not what we can achieve now because we have limits in sample size. So, and how big sample sizes we need in the future to achieve that limit? So this is what matters is this effect size distribution. So what I'm highlighting is 3 cancers: testicular cancer, oropharyngeal cancer, and breast cancer. And what I'm showing is that among these thousands of SNPs, again, the method produces an effect size estimates of effect size distribution. And again, I'm presenting in terms of the odds ratio, but presented in a kind of log scale. And so you can see that basically there are SNPs which can be positively associated or negatively associated. That's why some odds ratios are greater than 1 or less than, or could be protective. So it's kind of symmetric by design. But there is an important distinction between the effect size distributions we are getting across the 3 cancers. Notice that the testicular cancer has a very fat tail. And what does fat tail here means? That means this is a cancer, but there are larger number of SNPs with larger effects compared to, say, the breast cancer or oropharyngeal cancer. And that little differences, it may seem subtle differences, that makes a huge impact in terms of how big sample sizes we'll need in the future to improve the performance of the polygenic risk score for these cancers. So in the next figure, that's what we are showing. In the x-axis, what I'm showing is the sample size for genome-wide association study for training genome-wide association study, and again in thousands. So these are very big sample size we are talking about. 250 means 250,000, which is half of them will be case and half of them will be control. And y-axis, what I'm showing is the performance of the polygenic risk score that we can build using either of the 2 approaches. One is a very simple approach. That means you just focus on the SNPs which are reaching genome-wide significant, independent SNPs, or using this clumping and thresholding method where you allow for, instead of looking at very stringent threshold for selecting SNPs, you allow for a data adaptive threshold selection. Okay, so there are 2 different methods we are showing. And I'm showing that this is the theoretical expectation. Okay, and how does we expect this to change? And the blue dot is where the point where basically the sample size of the current biggest study. Okay, and that's where the polygenic risk score we could build at that time. And then the red dot shows what will happen if I double the sample size of the current study in the future, and the green dot if I quadruple the sample size in the future. And notice that it's very different for different cancers. In particular, testicular cancer, where we had the fatter tail, that means there are larger SNPs, or the larger effect sizes, they're going to move around the curve very fast. And I'll go towards the limit, which is the red dashed line, much faster, even with a 10,000 case and 10,000 control, which is the current largest study, we can already get an area under the curve about 75%. And with the doubling and quadrupling the sample size, we're basically going to start achieving the limit. So we don't need to do the 100, huge sample size US for this cancer, which is going to be very hard because testicular cancer is a relatively rare cancer. And then contrast that to the breast cancer, where it's a common cancer, where the sample size is already pretty large, which is about 125,000 case and control. And there the current PRS have actually kind of area under the curve can achieve is about 63%. And if I keep increasing the sample size, you'll see there is a slow growth towards the limit. So again, you can see that we have kind of come a long way, but after this, that if we keep increasing the sample size, there'll be only a gradual increase. So that the yield is going to be now slow. So you can see like the point here is that, first of all, across all cancer, we need fairly large sample size GWAS to make this polygenic risk score to be powerful, because the nature of the problem is that these are every disease is very polygenic. That means there are lots of SNPs, but each SNP has very small effects. And that's why to build these models, which is a lot of small effect models, to make it powerful, you need to first have very large genome association studies. But then there is a actual requirement can vary widely across traits depending on the underlying polygenicity and effect size distribution. So that's kind of on the, what I consider kind of the design of algorithm. We don't talk about, so much about, we talk about design a lot in terms of hypothesis testing setup, right? But we never talk about design when we talk about prediction. But this is, you can think about that, this is a design in terms of sample size calculation for prediction problem. But if you have a data, the more relevant question is that what algorithms do you use to build a polygenic risk score, right? So I talked about some simple algorithm. There's been a lot of work has gone on in this space, and a very simple description is that, again, a lot of those methods are based on summary statistics. So you can think about, if you have a summary statistics on millions of SNPs from a particular study, you can order the SNP from the smallest p-value to the largest p-value. And that's the left-hand side, that's what I'm showing. On the right-hand side, what I'm showing is a plot called linkage disequilibrium, which is basically what is the nature of the correlation between the SNPs. And so the first type of method was very simple. So they looked at the SNPs, which has reached a very high level of significance, typically referred to as genomoid significance. So we focus on those SNPs because we are very confident they are definitely associated with the disease after multiple testing adjustment. And we have to, using the correlation criteria, like you have to do clumping, because these are one SNP at a time analysis. So if you do not, you might double count, and that's why the people will generate these independent SNP using the clumping idea, but you basically sort them, and then any SNP which is correlated with a SNP higher in the list, you remove them, right? This way you keep the strongest signal and you remove independent, correlated signal. And then the next set of method was called clumping and thresholding. So people realize that if we just focus on the very top SNPs, we are probably missing a lot of signals, they are probably a weaker effect. They did not reach genome significant, but they are still contributing to the trait validation. So they propose that, okay, we relax the threshold. Instead of 500 minus 8, we make it more liberal. And we don't have to actually fix a particular threshold. We can vary it. We can, for each different threshold, we can construct a polygenic risk score using the kind of those set of SNPs selected according to the threshold and the estimated coefficient of the SNP from the same study. Okay. And then if we have an independent data set, we can try out those PRS and see which gives you the best PRS. And that would be the method called clumping and thresholding. And then more recent methods are, focused on that. Said, let's not do all this kind of ad hoc step. We can just go to we have good methods, in statistics using Bayesian or machine learning methods, which will allow you to build models, high-dimensional models, accounting for correlation among variables, parameter regularization. That's what we have been doing for a while. The only complication is that we don't have individual level data, right? We have working with summary statistic data. So people figured out that you can actually fit some of these models, like the Bayesian and machine learning type of method, like lasso type of method, using summary statistic data and some external information on the LD between the SNPs. And for example, just to see a flavor of different methods that are available, for example, even in the Bayesian setup, there are different methods depending on the kind of the effect size distribution or prior model they assume for the effect of the SNPs. So the first one, for example, a popular method is called LD-PRED. That is basically using spike and slab prior, if you are familiar with the term. It basically assumes there's a proportion of SNP has no effect, and the remaining proportion of SNP has a effect size following a normal distribution. The other extensions are, instead of having one normal distribution for non-null effect, we allow for mixtures of normal distribution, so that you allow for SNPs with a larger component of effects. Maybe there are SNPs which are much larger effect than other SNPs. We may need a mixture of different normal component. more recently, there has been an extension to, so that instead of discrete mixture of normal, you use a continuous mixture of normal. That means, again, if you look at the mixtures, basically you are allowing for different normal distribution with different variance component, right? With certain probability for each variance component. So the idea here is that you can use a continuous mixture on the variance component. That's called PRSCS, and there's also non-parametric methods have been proposed. We also recently, I had a colleague in my department, Aki Nishimura, and he's our local Bayesian expert, and he worked on, a lot of Bayesian computations, and and very, and also latest some of the latest Bayesian method. And he has been working on. He told me after, that he works on this prior called BRS Bayesian bridge, which I was not familiar with, and that has certain advantages, and they have successfully applied to a other type of large N, large P problem, prediction problem. And we said, okay, let's try it. Why don't we try? It's a new type of prior. Why don't you try in our setting? And so this is the specification. So basically, how the Bayesian bridge is specified. So again, J corresponds to the effect of a particular individual SNP in a multivariate model. And again, if you think about it, it's also a continuous mixture prior, right? So assume that every SNP has an effect, follow normal distribution with some mean 0, some variance parameter, it's called global variance parameter tau square, and a local variance parameter lambda, the global and local shrinkage parameter. The difference is, is how they're mixing. There's a distribution of the lambdas. Here in this case, they assume that it follows this alpha stable distribution, and the tau, the global shrinkage has a gamma distribution after the transformation. Now, what is the advantage of the method? One nice thing about this method is that you can look at after marginalizing over the local shrinkage parameter, the lambdas, the betas have a closed form prior. So you can see that the beta has now this form in the second line. And what is that? That is basically a generalized Laplace distribution, right? If I have alpha equal to 1, that is the Laplace distribution. And that kind of corresponds to the lasso solution. But here you have a alpha, alpha can vary from 0 to 2. So it allows for more flexibility, and you can try it as a tuning parameter. And also there's some computational advantage. That's why people use this in the other literature. It has because of this closed form nature, you can have better mixing in the give sampling for doing the MCMC, so things run much faster. So he said, okay, we'll throw it in the mix with the other ones and then do a thorough comparison. And so here I'm showing the performance of these PRS that are coming out of different Bayesian algorithms, and again into let's focus on a particular trait that is easier. Let's say this is called LDL, which is your kind of bad cholesterol. And we are doing this exercise in the UK Biobank. So the summary, the training data and the validation data, they're all in the UK Biobank. And so we are implementing different version of LD-PRED. Remember, LD-PRED is a spike and slap prior. Okay, but then LD-PRED, within the LD-PRED, there are a lot of details, like depending on whether because you have to, in all of these methods, you have to find a way to estimate the correlation in the SNP or the LD. But LD can be huge matrix, right? Because you're talking about millions of SNPs. So if you want to estimate a million by million matrix, it's very impossible. It gets very noisy. So you have to make assumptions, reasonable assumption about sparsity of that matrix. And you can make the different assumption. You can assume that the matrix is blocked or matrix is banded, right? And the banded means that if the SNP is far apart in the genome, they are expected to be uncorrelated. Or you can assume that the SNPs are, you can nicely divide the genome into blocks, and within blocks you have correlation, and between blocks you do not have correlation. And then you can decide like what would be the block size. The large block, we have contained two type of block. One is the like the large block, another is a small block. Small block on average contains 680 SNPs, and large block contains about 4 times, almost 4 times more than that. And then there are there are 3 methods we are comparing. One is the LD-PRED, the 2 version of it, whether the banded or blocked matrix with the LD matrix, the PRSCS, which is the continuous shrinkage prior, and the PRS bridge, which is the based on the bridge prior. And then you have 2 versions of it, whether the LD matrix was estimated from this 1000 genome reference sample, which is a relatively small sample size, and that is showing on the right side with a hyphenated bar, or on the left side, and the LD matrix is estimated based on a much larger sample size, actually based on UK Biobank itself. So what I main take-home message here would be like, one thing is like that we didn't see too much difference in terms of performance in terms of the prior. Is these practical details? Like for example, in general, we found that estimating the LD matrix with a larger sample size than the 1,000 genome reference sample people uses, it gives quite a bit boost in the power. So if you compare the bars in the left versus right, you'll see that on general, you get quite a bit of boost in terms of prediction performance. If you are stuck with a small sample size for estimation of the LD, then actually we saw that if you should use smaller block, because if you use large block, that means you are trying to estimate too many correlation parameters from a small sample size, performance actually drops. So you have to be it is better to use a smaller block size. And then between the methods, we saw generally one things are more comparable in terms of you are using the same data set for different sample size, same block size. We found that the bridge and the LD plate, the spike and slab, they perform more or less similarly. Actually, PRSCS in general performs slightly worse on average once you make the comparison fair, because there's a lot of unfair comparison in the literature, and the advantage of the method is not coming from the prior. The advantage of the method is coming from how they handle the LD. So that's something you have to keep in mind. But there are a few exceptions. So this is one nice example, IBD, which is the inflammatory bowel disease, where which is an autoimmune disease, and this is a disease where there are genetic variants which have very large effect compared to that there are things which are in the heavy tail. And this is where we start seeing interesting difference. You can see that the PRS bridge, which allows for heavier tail, nicely outperforms the two other prior, which in this case, LD plate 2 and PRSCS, with everything else being equal. So this is where you start seeing sometimes the prior can matter for few traits where you expect might be few variants, which has a much larger effect sizes than the other. So that's sort of quick summary of what happened in the original kind of the PRS development method. But more recently, the focus has been to this generalizability problem of this polygenic risk score, because most of the studies to date has been based on the European sample origin samples, and we have very little or almost no study sometimes from other ethnic groups. And then as a result, if the polygenic risk score we are getting, they perform relatively well, in those populations, the European ancestry population or origin populations. But if you take into some other populations, particularly in African ancestry populations, which are genetically and genealogically the farthest, the performance really drops. And so as a result, if you think about this polygenic risk score, if we prematurely take it to clinical application, then you can see that, yes, we'll find maybe in the European population, we'll find individuals at high risk who would be recommended for certain type of interventions. But if we implement that in other population, it's not going to give a lot of spread of the distribution. I'll find very few people who will meet those kind of criteria. And so that can generate also inequity and exacerbate the current inequities. So, and the main reason people have identified that, why the polygenic risk score generated from one population does not generalize to other population. One is that the big difference is the allele frequency. So currently, the genome-wide association studies we have done, we have designed for European populations. So basically, we are studying variants which are more common in European populations. We have identified causal variants which are more common in European populations. But those same variants, even if they have the same causal effect, they can have a very much lower frequency in another population. And as soon as it becomes lower frequency, even if the causal effect is the same, the predictive power in terms of how much variation of disease risk it can explain, it drops. So this is the most important cause that the why PRS does not perform well in other populations, is the allele frequency differences. The other big problem could be differences in the correlation pattern. Right, in the European population versus African population, for example, because African population is older, there are less correlation between SNPs, and there is more recombination events, and that's why there are less correlation among SNPs. In the European population, even if we do not get a causal variant, if we get another variant which is tagging the causal variant, and that is included in the PRS and is performing fine for prediction, but the same variant is not going to perform well in another population because it's not the causal variant. and it does not have the same correlation with the corresponding causal variant in another population. So you lose the prediction power that way. And finally, it is possible that even if you could identify the causal variant, their actual effect sizes are different because of the different population might have been exposed to different environment. If you believe in gene-environment interactions, then that it is possible that the effect sizes are also could be different. So the latest methods have tried to do is that, like how to, kind of using that of existing data, how can we improve the prediction performance in the non-European populations? You can think about like kind of various type of transfer learning approaches, to develop better polygenic risk score in non-European population or minority population where the data sets are still quite small. So the first set of ideas was very simple, like this kind of weighted method. So the idea is that if you have two sets of data, let's say one for a European origin population, where you have much larger sample size, and then a smaller data set, which is maybe in a target population, like an African ancestry population, where you're trying to do better. What you could do, like you can take one of the method, apply them separately to the two separate data set, and you get two polygenic risk score. One is for the European population, other for the target population. But now, because we also know that although these populations are different, we do expect that some of the genetic variants will have similar effect, right? So the one way of borrowing information is that we said, okay, now let us combine the two polygenic risk score in a linear using a linear combination that will optimize the performance in the target population. And so if you need a little tuning data set to estimate the weight, W1 and W2, based on the tuning data set, and that way you can borrow the information on the larger population to improve the performance in the smaller population. Very simple idea, actually works pretty well. But if you're a Bayesian, you want to do more in an advanced way. The obvious thing would be to now think about, okay, you have a effect size distribution we talked about for SNPs in one population, but now you can talk about the effect size of the SNPs across population. So what will you do? You'll propose a multivariate prior, right? Like instead of a univariate prior, you'll propose a multivariate prior that will allow to specify some correlation structure that says that, yes, if a SNP is a causal in one population, it's also likely to be causal in another population. So based on that idea, there are different methods. XPath is a method which basically assumes multivariate normal for effect size distribution. And with the multivariate normal, you can easily specify correlation structure. PRSCSX is the extension of PRSCS, the continuous shrinkage prior, but again, to multivariate setting. And then we have been recently playing around with a couple of methods, simple-minded method that can potentially improve. One of them is city slaves. So I'm going to give you just a quick overview of that. And I'm sorry, this slide is a bit busy, but just hang on with it. So the idea is that suppose you have two sets of summary statistics on the top. What I'm showing that think about the two columns is the one set of p-values from the European population, and which has more stronger signal, and the other is from a maybe a smaller data set from the African population. And we wanted to do an extension of clumping and thresholding, but to do sort of this idea about clumping, that means generating independent SNPs, you have to define some rules, right? Because we are now working with two sets of population and they have different correlation structure. So we said, like, if for a SNP, we will check whether the p-value of the SNP is smaller in the European population or is it smaller in the target population, which in this case, the African population. And if it is smaller in the European population, which is a stronger signal, then we'll use the European LD estimates to do the clumping step. Or if it is smaller in the African population, we're going to use the African LD estimates for do the clumping step. And then for the thresholding, what we do is that we propose this idea called two-dimensional thresholding, where we allow for sort of this threshold, two different set of threshold, one for the set of p-values for the European and one set of threshold for the p-values for the African. And the idea is that we want to allow different threshold because we know the African sample size is much smaller, right? We expect the p-values will be weaker. So if we allow the same stringent threshold to be applied for both European and African, a lot of the time they will not meet the criteria in the African population. So we said, let us make those thresholds separate so that it is possible we can include a SNP which was very strong signal in the European and some weak signal in the African, right? And that would be a good candidate to be put into that African PRS that we are trying to generate. And so that we allow for different threshold, but we do not pick a fixed threshold. We vary it over a set of grid point. Okay, so that the first step shows that, okay, how do you select SNP in the algorithm? And now once you select SNPs, then you have to say what coefficient do you attach to these SNPs? And remember, we are trying to develop a good PRS for the target population, which is in this case African ancestry population. But if we only take the estimate of effect size from the African ancestry population, that would be less efficient, because we do know that the effect sizes tend to be similar across population. So what we do here in this step, we do a very simple empirical based method based on a normal multivariate normal prior, which comes in a closed form, right? And if you want to do here we kind of simplified. We said like for the selected SNP, the SNP effect sizes follow a multivariate normal distribution, and from that you can kind of do some simple empirical based set of calculation for coming up with a better estimate of the effect size in the African population, which borrows information from the other populations. And then finally, we add this called super learning step, which is usually in this kind of thresholding idea, you try multiple threshold or multiple tuning parameters, and then at the end you pick one optimal tuning parameter, right, in the independent data set, and see which combination of tuning parameter gives the best performance. But instead of doing that, we say that let's take all the polygenic score we get for different combination of threshold and then put them in another model, and this is called the super learning step. And this has been existed in statistic literature in a while, that you can take algorithms from different types of algorithms, and then at the end you combine them with another algorithm. So that's what we are doing. We take all those PRS, and then at the end we run it through a final step, so that we can come instead of picking one winner. And this is sort of shows that the performance of the different methods in a particular data set. Actually, this is the collaboration we have with 23andMe. And as you may know, that 23andMe has actually possessed some of the largest genetic database. And so we are very lucky to be able to work with them. And you can see that some of the sample size, for example, in European, and these are prediction for cardiovascular disease. And you can see the sample sizes are huge. For European, they have more than 1 million case and 1 million control. But even for some of the non-European population, their sample sizes in tens of thousands of cases and tens of thousands of control. And the way the collaboration work is that they split the data into 80, 10, 10. The 80% was used to generate summary statistics, and that was handed over to us. And then we apply our method. We generate a lot of PRS. We send them back. They tune with the 10% of the data set, and to select and this kind of weighting and all that. And then finally, in the last 10% of the data set, they report the final performance of the model. So these y-axis is showing the final performance of the model in a completely independent validated data set. And if you look at the different methods, so the blue ones in the single, what I call single ethnic method, which is basically you say like, okay, in this data set you can see that if I want to predict CVD in African population, they already have a pretty large data set, like 47, almost 50,000 case and 50,000 control. What would happen if I take one of the standard algorithm like clumping and thresholding or LDPRED2, just and develop it only using that data? Don't forget about the European data. The next set of method, not the method, the green ones, the shaded different shades of green, we said, okay, here the European data set is so large, I can develop a very good PRS. And maybe that PRS will also perform as well in the African population. So that's showing that what will happen if you take the European PRS, it could be either clumping or thresholding or the LDPRED2 method. How does that perform in the other population? The next one is the multi-ethnic method, where you actually borrowing instead of using one versus the other. The first one of them is the weighted PRS, that you generate the individual PRS and then combine them with the weighting approach. The one after that is the PRS-CSX, that's the Bayesian method. And then the last two set of method is this new method we propose, the CitySlab. There are two versions of it based on whether you combine the 5 ancestry data all together, or you just combine that particular ancestry with the European ancestry and to analyze 2 at a time. So let's focus on we're mostly doing 2 ancestry methods. And generally, over the picture, you can see that first of all, you can do a lot better than the single ethnic method. And you can also do a lot better than at least some setting, for example, in the African prediction, in the African American, you can do a lot better than the weighted method. But among the newer method, I don't think there is a clear winner here. So, depending on what population you have, what trait you have, it can vary. For example, in African population, we do see that some advantages of the CitySlab method compared to the PRS-CSX. But if you look at the East Asian population, you see that the PRS-CSX is doing a little better. So I would say that overall they are comparable, but there are situations where depending on the trait and the population, one can be better than the other. So there is no universal winner here, and that is the general story here, that there is no universal winner. Yeah, it depends a lot on the, as I said, like it really goes back to the performance of the genetic architecture. That means, for different, for the same disease, different population, there might be, different effect size distribution, number of causal variants might vary, the LD is different, right? So all of that actually affects the performance of the algorithm, right? So in one setting, it prefers one algorithm because in that algorithm, whatever assumption is there is more suitable for that population, right? So that's why, because this heterogeneity is there, that's why we are seeing that the relative performance can also vary a little bit. So there's another method that was developed recently by one of my students, which is, so one more thing I forgot to mention. So one of the big advantage of this method compared to the Bayesian method is the computational speed, right? Everything is closed form. The method that I showed is basically, every step is in closed form. So you can see that the computation is extremely fast compared to the Bayesian method. And so the fact that the method, at least overall performance, is similar to the Bayesian method is, I would say, quite impressive. And then there's another paper we recently worked on. This is called Prosper. So we wanted to see, we were encouraged by the performance, the good performance of a simple method. So we said, okay, now can we kind of extend it to like really do more rigorous method using a penalized regression framework? And what we did was what we call the method is called Prosper, which is stand for polygenic risk score based on ensemble regression, lasso type of regression. So here is in the middle of the slide, what I'm showing is the loss function, for when you're trying to develop a PRS in a multi-ancestry setting by analyzing them jointly. And there is sum I equal to 1 to n, which correspond to m ancestries, right? And the first part of the first two term is basically correspond to your loss function, like the usual loss function of the, for example, if you're writing for a continuous rate, but represented in terms of summary statistics, right? Because we are dealing with summary statistic data, so you can write down your loss function in terms of summary statistic data. The last two terms are about the penalties, right? So you can see the first penalty, we have the L1 penalty. And for each set of beta, there is a set of beta that we are trying to estimate for the SNPs, and each one set of beta for each ancestry group, right? And I'm kind of putting a L1 penalty on each of them, and then there is finally a reach penalty, which is basically kind of trying to shrink the parameter from different population to be more similar, right? We want to penalize, and so if they are different, we want to put a penalty. So because we think that the effect sizes have some similarity across populations. Now, if we just want to fit that model, then what happens is that there are a lot of tuning parameters, because for each population, you can have one tuning parameter for the lasso, and then for the ridge, also for each pair of population, you can have different tuning parameters. So we had to simplify the problem for computational purpose. So what we did was that we said, before we do this, first we'll do some pre-processing. And the pre-processing will be that first we're going to analyze each ancestry data by themselves. And there is a method called lasso sum, which basically fits the lasso to data from single ancestry. Okay, and once you do that, you get an initial estimate of the tuning parameter for that population, the lambda parameter, which is called a sparsification parameter. And then we feed it into and then we take those and we plug that back into our joint estimation. And so this lambda I0, those are coming from the initial analysis for one population at a time. And then we say that, and then we just allow for one more parameter so that we do not be too rigid about that, whatever comes from the initial value. We just allow a single penalty parameter and scale it by the initial estimate of the tuning parameter that comes from the single ancestry analysis. And then we allow for each pair of population, the amount of similarity is kind of similar. That's why we have a constant C, and it is not pairwise specific parameter. So that way, at the end of the day, we just have two tuning parameters: the lambda and the C in the whole functions. And for each lambda and C, this can be solved in coordinate descent, so very fast. So you can solve these things in a extremely comes out as a very nice solution. Things are very fast. And then finally, again, use the idea about the ensemble regression. For each lambda and C, we can generate a corresponding polygenic risk score. And then instead of taking the optimal, like we have a tuning data set, and based on which we could have picked an optimal lambda and C combination that gives the best performance. But instead of doing that, we kind of take all the polygenic risk score for different combination of the PRS and then fit through an ensemble regression method. So this is the kind of the expanded analysis now with a kind of more trait from 23andMe, also more method. And again, the last one is basically the same method as before, but then we have added the Prosper, which is the last box. And as you can see that this kind of method can also give better performance. Generally, we found that these methods tend to be the best or close to the best. And sometimes it can give, especially in the African population, you will see that it can give notable improvement compared to the PRS CSF or even the CT-SLED method. And some other data set, like, you see even more impressive performance sometimes because this is the lipid traits. Again, here you can see generally it tends to be the best. Of course, there is sometimes the second best method is also as good, but overall this was remained close to best or sometimes better than the next to best method. And this is also good because this method is in terms of computational demand, it is similar to the CT-SLED. It's much faster than the Bayesian method because all things are like almost comes out as a closed form. So one of the questions we asked, so one thing you do notice that here, for example, that even with a lot of fancy and better methods, if you look at the performance of the models in the African population, they tend to be lower than the other population, especially compared to the European population, which is not there. They are more like, if you look at the Latino population performance or South Asian population performance, they are more comparable to the European population performance. But consistently, we see like sometimes the African population, the PRS performance, even with better method, can be lower. And so we asked the question, how large sample sizes do we need to have in the future to make this? It's a sample size issue, right? That it is not just better method, we also need to collect more data, right, on those understudied population. So you can, now that we have some good method and some of them are very simple to implement, so we said let's do simulation, to see that how big studies we need in the future to kind of close the gap. Okay, so here is the simulation, and this is a forward simulation. That means we actually simulate genotype data using realistic haplotype patterns, using patterns observed in 1,000 genome type of reference data set, so that they match the allele frequency linkage disequilibrium pattern pretty close to what is known about the these things in the different populations. And then we have a scenario here. So suppose that we have a large genome-wide association studies for European, about 100,000, and then we are kind of trying to have for the other ancestry population, we are trying to build the genomes over time, to make it larger and larger. And the performance, the red dashed line showing what is the performance we observed on the European population PRS built based on that European data. And the point is that we want to be close to that red dashed line. So you can see that for the African, American, East Asian, and South Asian population, you can see that we can basically, with almost half the sample sizes of the European sample sizes, and by borrowing information the European, we can basically start getting similar performance as the European, right? So that means that we do not need to have as large a sample size as the European data to get performance as the European, because we are going to have somewhat larger sample sizes than currently, and we're going to borrow information from the other population, and that way, then we can start matching the European performance. But we did observe a consistently, underperformance in the African population, and this is a unique setting. So because you have to make it, when you do simulation, you have to make a lot of assumptions. And one of the big thing about the way the African ancestry population is different than the European is that African population has many more genetic variants, like almost double the other population. And what happens is that in this model, we assume that the amount of heritability that could be explained by common variant, they are the same across the different populations. And as a result, so if in the African population, there are twice the number of genetic variants. So as a per SNP level, the average effect size or the average amount of heritability explained is now suddenly half. compared to the other populations. So in that case, and that is a very key parameter, how prediction will perform, because in this particular simulation setting, that average heritability per SNP is half compared to the other population. We see that there is always a consistent underperforming. But on the other hand, if you believe that actually because African population has more variance, so it might have proportionately more variance, in terms of heritability explained, then things will be more comparable. So right now we do not know where we are. That is it, the African population, they have more genetic variations, common genetic variations. Does that mean that they will have more heritability for these traits? Or does it means that the average heritability is small? And so we do not know, so we need to collect more data and do this kind of analysis to see what kind of sample size requirement we have in the future. Maybe in reality, it will be somewhere in between. And one more thing we studied in the simulation is that like how big SNP content has to be, because Africans, for example, if you think about the number of SNPs we have, and they're up to 19 million common SNPs. But do we really need to include all of the 19 million SNPs in these algorithms to keep improving the performance? Because there is a lot of correlation in the genome. Maybe if we include only 1 million SNP, that will be enough to have the performance, because handling 19 million SNPs in this algorithm is pretty complex. What we found is that if the sample size for the training data for the African population is large enough, like 80,000, you keep actually seeing a gain. That this one is based on about 1 million SNP, and the second one is that a common set of SNP called half-map SNP. The second one is called a set of SNPs carefully selected to represent genome in diverse populations. So it's a half-map SNP plus these SNPs in the mega platform. This is a particular chip, and that is a total about 2.8 million SNPs. And then the final one is like all the SNPs that are observed in 1,000 genome, which is about 19 million SNPs. So you can see like that we found that as long as your training data set is large enough, you actually keep getting increase in performance with more larger SNP content. That means that in the future, models, whatever advanced algorithm you're trying to develop, they should be able to handle this kind of scale of variance. If we just prune it ahead of time and reduce it to 1 million, because that's the algorithm can handle, they're intrinsically going to lose information. So that's why scalability is going to be very, very important. So that's kind of your take-home messages from this part, is that after a lot of careful work, a lot of evaluations through simulations as well as data analysis, we didn't find any method is uniformly powerful in all settings. And because of that issue I mentioned, because there are different scenario, different population, different trait has different properties. So so I think this kind of ensemble type of method that you generate PRS across multiple methods, and then at the end you put them in combination, and that will have might be a good approach to go in the future to develop the best PRS. And as I said, that some intrinsic challenges, beyond algorithm, beyond sample size could be there, especially in African ancestry population. And the other thing is like the algorithms need to have very scalable to be able to handle large SNP contents. Okay, that brings me back to the last part of my talk. I'm going to make it very quick about it as we are running out of time. So there is a lot of debate about, okay, we can develop this polygenic risk score, the fancy algorithm, but what would be their potential clinical utility? And this is where I think, it's really important to think about the way the risk information is communicated, and the best communicated is in terms of absolute risk. Like if you have somebody's profile, genetic profile together with some lifestyle factors and stuff like that, or other biomarkers, what we need to have models that can predict an individual's risk of developing certain outcome over absolute risk over a future time period, because those are the things most clinically understandable. And that's why we need to kind of bring in all the information of the PRS with other factors into developing this absolute risk model. And that has been a lot of focus of our research. So when you're trying to show clinical utility, of these things, for example, here also, like in Fred Lee's work on colorectal cancer, they take similar approach. Like they try to take the polygenic risk score, combine it with other things, and then show particular clinical application. Like, will it help, for example, to decide like who should start colorectal cancer screening and at what age? Because there is a particular intervention available. You can think about in terms of what absolute risk threshold people should start doing the screening, and then you can start showing clinical utility. So these absolute risk models are very critical. So one thing I'm going to emphasize, there's a current emphasis, like people are showing this performance of this polygenic risk score, and in a disease-specific way. Like, I showed application breast cancer. Lee has been showing application colorectal cancer. There have been shown people are showing application prostate cancer. But one interesting thing about this polygenic risk score is that they're a pretty unique technology. If you think about it, all you need to do is to collect your DNA sample once in your lifetime. And using that DNA sample, you can collect polygenic risk score or compute polygenic risk score for all kind of outcomes. Like you can do the 15 cancers or 10 other major diseases, and also BMI and some other, your tendency to smoke more or alcohol, drink more, everything you have a polygenic risk score now. So one thing, and so people are trying to show that the cost benefit of this kind of genetic data collection and score collection. So I think like we also need to show, other than showing disease-specific utility, we need to show that their broader utility, because this is a multi-disease, multi-cancer technology. And so we have, for example, shown with the work with Alison Meissner, who is currently a member in Fred Hutchinson, like you can take a polygenic risk score for different outcomes and risk factors and put them in a model to predict all-cause mortality, to show a broader utility. So one thing, so just on the same path, so we just did a little bit of work on this, like how this polygenic risk score, which is a multi-cancer technology, can come together with another multi-cancer technology that are being, there's also a lot of excitement about it, and is the multi-cancer screening using liquid biopsy. So if you're not familiar with, this is a technology, the promise is that, it's a liquid biopsy, which means that you can think about you are having a biopsy without actually taking a tissue sample. You can take a sample from your blood, which is very less non-invasive, and that can potentially detect tumor DNA, that if there's any tumor in your body which is still not manifested, that you have a diagnosis, but that blood sample can detect traces of DNA. And that is and there's a lot of new technologies developing to do that well. And so there is a lot of excitement and sometimes hype about this technology, but a lot of different institutes investing a lot of money. But if you look at the performance of some of these tests, the positive predictive values are still low. That means if you have a positive test, what's the actual chance that you have a cancer? That probability can be still low. And think about that is kind of, it has a lot of harm associated with, because if you have a positive test, you do not know where you have cancer, right? You have to do MRI, CT, and that has cost, that has psychological harm, and you'll have to go through all those kind of things. So the idea would be to, even if these tests were one day were shown to be that they're ready to be used, you're not going to give it to everybody. You're only going to give it to people who would be probably at a certain level of risk for which the positive predictive value of this test is going to be high. But this kind of idea has existed in cancer screening a long time ago. For individual cancers, you propose that, okay, you will do screen if the individuals at a certain level of risk, right? But in the multi-cancer context, where you are not cancer specific, so how do you think about identifying individuals at risk, right? So that, and I thought that this is where a polygenic risk score could be useful, because it's actually also a multi-cancer technology, right? Because I can have a polygenic risk score for breast cancer, I can have a polygenic risk score for colorectal cancer, I can have a polygenic risk score for many of these cancers. And so this is what we recently did. So we kind of developed a validated model to predict risk of at least one of 8 or so common cancers in men and women separately, based on a few basic risk factors like age, BMI, smoking history, because these are things which affect many cancers, and these are across cancer. Family history of a few cancers, common cancer, and this polygenic risk score for these individual cancers, and which we could download information that are available from public data sets. And then we built a model based on this UK Biobank and a part of the UK Biobank data to predict that this combination of risk factors, how well that can predict an outcome, which is a composite outcome that somebody has developing a cancer of this out of these 8, and that's what your outcome was. And then based on that, we can define risk level. Like, what is the risk if you are developing at least one cancer in the next 1 year, if you have certain age and a combination of this risk score, right? So I'm not going to go to the detail of that. These are some of the cancer. These are the number of SNPs that was included in some of these cancer and defined the polygenic risk score. This is some of the details about how the model was developed. But that is the basic, Cox model that we use. So you can look at the time to event outcome. The event is the first cancer incidence. Okay, and then that's what we are looking at, the event. And then the T is the age. That's where we put the age. And then in the Z's are the risk factors like BMI, smoking, family history, and the PRSS. And we developed an absolute risk model. And this is what I'm showing, the 1 year absolute risk for women in this case. And according to 3 models, one is like if I do not include the polygenic risk score at all, just use the age, BMI, smoking, and family history. Another is just based on the polygenic risk score. Another is the combined one. And on the x-axis, what I'm saying, how the risk changes as a function of age, right? And we all know that cancer risk increases as age increases, and that's the gradient you are seeing. But you also see a very strong gradient on the y-axis, and that is the gradient correspond to the percentiles of this, what I call pan-cancer risk score, which is the risk score defined by the combination of the polygenic risk scores and the other few risk factors, right? That defines a risk score, and I can divide up people into whether you're in the top 1 percentile, next 5 percentile, and so on. And what I'm showing is the actual level of risk that is observed in those categories. So you can see that there is a very strong gradient of risk, more so than the risk gradient you see at the age level. So that means that this does give quite a stratification, and the combined model does better than just if you look at either the polygenic risk score model or just the other model. Now what we did is that, so these are the risk level we can achieve. Yes, 2 minutes. Yeah, I'll finish. And so now what we wanted to calculate is this. Suppose I want to now apply a test, okay, this kind of cancer detection test at this different risk level. X is a particular risk level, and I want to find what is the positive predictive value of the test is going to be. So if you apply the basically Bayes theorem, then you can see basically it boils down to you have to know the sensitivity and specificity of the test at that risk level, and then what is the risk level, the PX, which is the risk level. And then there are studies now that publish the sensitivity and the specificity of the test. So we actually import that from the literature. I know the actual level of the risk from just what I showed. From that, then now I can calculate what is the positive predictive value of this test is expected at the different risk level. And this is what, again, for female, for one of these popular tests, which is called Gallery, which has been recently evaluated in a large study in UK. And we have taken the sensitivity and specificity of those tests, which are quoted before. So if you look at the last column, which is basically show the positive predictive value of the test as a function of age of individuals and the level of risk that is defined by this pan-cancer risk score, you can see there's quite a bit of variation you can get. Like, that, for example, if you want to make sure that you only apply this test to individuals who have at least 50% positive predictive value. So you can see that there are people, you have to be fairly old, like 60 and above, and then at a pretty high level of and you have the top 20% of that pan-cancer risk score to get to that threshold. So you can see that this kind of risk stratification might have some value to decide that who should be given so that they this tests are effective for them. So just the final concluding slide is that for showing clinical utility, the absolute risk models are absolutely essential. And then and also, like another point we try to make is that other than showing clinical utility for individual diseases, individual cancer, we can try to do things to show more broader utility in terms of, especially if you're thinking about, is this a cost-effective technology, right? Because the nice thing about the technology that once you collect the data, you can distribute it over many, many different diseases. So this was one application to show the broader utility. With that, thank you for your attention. And I have many people to thank, but I will not individually mention, but happy to take questions.
Yeah, we have some time for a few quick questions, maybe.
Okay.
People online can also post their questions by muting themselves.
Other chats.
Any questions? Yeah.
Yeah, very impressive. So, the question is this: to really share us for like individual populations, European standard, but especially this one, we have a lot of evidence. Now we developed first of all, how do we want to find a list of evidence? And we have this score as a following.
Right. No, that's a great question. I don't think we know the answer yet. There is a lot of thinking is going on behind it in various, for example, primate consortium. There's a lot of discussion around that topic. And I think there is a more and more consensus now that we need to start getting away from this concept of categorizing people based on self-reported ancestry. And so one of the idea would be future would be to generalize some of this method where switching the self-reported ancestry by more genetic ancestry, like so you have to use like PCs and things like that, where you can take into account the continuous genetic ancestry information. But one thing is that I think that's how it should go. Like, and I think some of these models can be generalized. You can think about when you allow for heterogeneity of the SNP effects, right, by this categorical ancestry, you can replace that by interaction term with a genetic ancestry, right? The continuous variable, right? That's possible. So theoretically, you can extend the model. The one big challenge is that a lot of the data is still available in the summary statistics form, right? But if you want to develop this kind of PRS, which accounts for continuous genetic ancestry, you actually need individual level data, right? So that's something we have to solve in the future in terms of data availability. People have become so comfortable with working with summary statistic data, they almost do not want to deal with individual level data anymore, right? But now we are going to be that, but if we are to go away from that categorical definition of ancestry and work with the admixture component or PC component, we need to start having access to individual level data. So there is some, I would say, logistical issue, just not methodological issues here.
Any other questions? Yeah. Yeah.
Yeah, so that's a great question. So yeah, so there are some hint now, for example, diabetes, people have shown that there is based on especially the rare variants from exome, they can contribute to the risk prediction. And there's another paper I saw, I think not a paper, it's just in archive. They showed that in the common variants, the regions, if you look at the rare variants, and they can find people like who are really extremes of risk, right? So I feel like very rare variant. These are, we are talking about extremely rare variant, so not of the intermediate frequency. They have potential to in the exome, not the whole genome. I think that's pretty noisy still. But in the exome, rare protein coding variant, they have further some potential to improve to be included in the future in a polygenic risk score. And there is some early hint of that. There are a couple of papers I have seen.
All right, I think we are basically out of time. So let's thank Dr. Chatterjee again.